see the derived distribution. So I showed you before the slide, and I told you here now we have a tether and everything is cool, but we have to actually validate that everything is okay with our measurements, and we have a correct DNA that is tethered according to what we think. So we have 3 criteria of checking this. The first one is to look at the scattering plot. And the scattering plot should be round due to the nature of the random. The second criteria is to look at the fitting, the goodness of the fitting of our data, and you can see in the dot plot is the exponential, experimental data. And the next criteria is to look at the persistence, which is according to the literature about 50 nanometers. So if all of these criteria are fulfilled, then we move on and we introduce the protein to the solution. Here's some measurements we took with the HU, and you can see to your left, these are of course scattering plots, and you can see to your left, blue represents more flexible. Now let's take a look at the right plot. And at the right plot, again, the relatively low HU concentration is shown in red, however, high concentration is shown in blue. And you can see that this time the trend is opposite, because now the beta actually is used in larger volume. So the end-to-end distance is bigger and the DNA is less flexible. And we also present in front of you the 2D distributions for HU, for different HU concentrations. When we start by no HU, and we see that persistence is about 50 nanometers. Then we introduce 100 nanomolar HU concentration, and you see that the peak goes to the left, the distribution is narrower, and then the persistence decreases to about 40 nanometers. The trend goes on, and you can see for 1200 nanomolar, the persistence is decreased to about 25 nanometers. And now when we go to the 1000 nanomolar, so now you can see that the trend is opposite, and now actually the persistence is increased to about 35 nanometers. The black. I didn't. Sorry. Forgot. Okay, so here's a plot summarizing the dependency of the persistence in the concentration of HU, and you do see the bimodality where at first the persistence is decreased and then it's increased. Not that it was installed, but still increased, and you see the bimodality. And we also wanted to look at the structures of the HU DNA complexes. So this is why we did some AFM experiments. And you can see the cumulative distribution function data. And here's up to 15 degrees. When we use no HU, already 70% of the angles are counted. But when we go to a low HU concentration, less than 65% of the angles are counted. And that actually made us conclude, it has to be because there are more high angles for the relatively low HU concentration, and that implies of a more flexible DNA for the low HU concentration. And then when we go to the high HU concentration, we see that already 73% of the angles are counted at this low angle. So that means that the DNA is less flexible for the 1000 nanomolar. But we also did, proposed Gaussian fitting to the CDS data, and for the no HU concentration and the low HU concentration, one Gaussian was very good. However, for the 1000 nanomolar, we had to do a 2 Gaussian fit. And that actually implies on 2 populations. One of them is of small angles, and the other one is of high angles. And that led us to conclude that HU induces incoherent persistence along different regions of the DNA. So the DNA has different flexibility along different regions of it. So you can't actually talk about persistence now. Just you can say it's an average persistence, but it has different flexibility along the DNA. And we wanted to compare our results to previous studies, and you can see in plot A the Stavins experiment that was done by Sagi, and you can see first you see the bimodality where first the transfer efficiency is high and then it's low. And in plot B you can see Vanut experiment with the magnetic results. We also see the bimodality when the persistence is first decreased and then it's increased. And you can see that in Vanut experiment the persistence actually increased up to 150 nanometers almost, which is about 3 times higher than with no HU. And in our experiment, you can see that there was strength, again, the bimodality, but it just increased up to about 35 nanometers. So we actually try to answer these differences. And we have 2 answers for that. The first one is the biological answer. We use the BST bacteria, they use E. coli. And the second one actually goes from the different nature of the methods. Well, they use magnetic tweezer where you stretch the DNA and you actually might alter the interactions between the HU and the DNA. We just use the post-free technique. Therefore, we look at the nature of the interaction without perturbation. So we also wanted to find a physical model to the B functionality of HU. This is why we adopted the model of Radford and Rabin from 2008. And you can see in plot B that when one protein occupies a segment, it bends the DNA. However, in plot D, when 2 proteins occupy neighboring segments, they actually lower the spontaneous curvature and it makes the DNA be more stiff, more straight. And for example, you can see in plot E a typical chain, when you see that in different regions of the DNA, there's like different flexibility. This is the different amount of proteins that are bound to the DNA. And to the right, you can see an illustration of a typical experiment in the TPM setup. You can see that, for example, when one protein occupies one segment, then there's a bend, and where there are 2 proteins occupying 2 neighboring segments, there's like a straight line. Just for an illustration. So, we thought to ourselves what could be the benefit of such a bimodality behavior in biology. So, who helped with the TPM protocol, Noam and Gil from Science project in molecular biology, all of the Bar-Ilan members and all the students from Bar-Ilan.
I want to thank Guy for making it so compact. Questions? Yes, Ray?
How large is the gold particle?
80 nanometers.
80. And how large is it?
About 925 nanometers. So, I don't know if you're thinking about the size, so you have to think, of course, of low Reynolds number. And if you're thinking about conformations, then there's a criteria which is called the exclusion number. And this was just by a paper that was published by Nelson, and we are really under this limit when the dig is too high. So in our case, this is what we were thinking.
The gold particle by itself is also probably negatively charged, right? So proteins are close to the gold particle.
Negatively charged because of the gold.
Gold,
So much.
probably they have to functionalize, stabilize it, things like that. It has to be charged, otherwise it will be stable. So if it's negatively charged, you also have protein absorption to the gold particle itself, right?
But it should be much smaller than the violence to really make an effect. You're talking about making effect with the DNA?
Just asking about modification, whether you have excessive absorbance of the protein itself to the gold particle.
Oh.
Is that the reasonable scenario?
I didn't check. I can't check this. I mean, I don't think so. Just because even though there's still, well, it doesn't make any. I don't think that it should be tethered or linked to absorb to the protein, because the other phenomena it could be also absorbed to the surface, but because we actually tested it, but I didn't check it, but I don't think it's actually the case, because otherwise there will be some unspecific value to the gold nanobit, and you will see actually fluctuations in the intensity. So I would doubt it.
This is video correctly?
We use the CCD. We just we do like a not video, because we take a set of pictures in every time spot.
What is the rate?
We use like with 100 phone 100 hertz to 300 hertz.